So far, a lot of work has been done to achieve a net gain of 4 ATP molecules, but a lot of potential energy has been stored in the NADH and FADH2 molecules. The big energy payoff is in oxidative phosphorylation, where the energy stored in those molecules is used to create 34 ATP molecules. OP consists of the electron transport chain in chemiosmosis. The final step, oxidative phosphorylation, consists of two parts, electron transport chain and chemiosmosis. Electron transport chain involves the movement of electrons in the presence of oxygen to work. NADH and FADH2 store energy and release it in the form of high-energy electrons, but they cannot do it if O2 is unavailable. Oxygen is an electron acceptor. The high-energy electrons cannot flow unless they have a final destination. Oxygen is that destination and will take the electrons. High-energy electrons donated by NADH and FADH2 molecules move down the inner mitochondrial membrane and are accepted by oxygen through a series of reduction and oxidation reactions. As the electrons move down the membrane, they transfer their energy to protons, moving them across the membrane. There are now more protons outside the membrane than inside, resulting in a net positive charge outside the membrane and a net negative charge within. The outside protons are attracted to the inside, but the membrane only allowed them to move out. ATP synthase provides a channel back to the inside, and the protons move through it, active transport. The final movement of the protons uses their electric potential energy to add a phosphate group to an ADP, creating ATP. Electrical energy is converted to chemical energy. The ETC in chemiosmosis is the same process used by photosynthesis. The electron energy was transferred to the protons and finally used to make ATP. Oxygen takes the de-energized electrons and protons, making water. The movement of the protons is called the proton pump. Depending on the sugar substrate, cellular respiration can occur at different speeds. The graph shows that maltose and dextrose elicit a significantly faster respiration rate in yeast than other sugars.